Thank you, everyone, for attending Chardan's leadership call with President and Chief Executive Officer of Synlogic Therapeutics, Dr. Aoife Brennan. My name is Keay. I'm one of the Senior Healthcare Research Analysts at Chardan, where our vision is to help identify companies with high potential for investment returns based on creating real value for society through disruptive innovation. Before we get started, our compliance department has asked us to read this statement. Our participant has attested and agreed that they will not share or discuss any material, non-public, or confidential information that they are aware of. A recording of this call will be made available to clients after the call. The format for this event is a 15-minute presentation followed by about 15 minutes of Q&A. As a reminder to attendees, if you'd like to ask a question of our speaker, please type it into the question box under the video player, and we'll get to it if we have time. So with that, Aoife, thank you so much for joining us today. Why don't you go ahead with your presentation?
Great, and thanks, Keay, for hosting us. It's always a lot of fun to tell, to inform everyone of the company updates and the exciting year we have ahead of us. So just to remind everyone, I will be making some forward-looking statements today, and I would refer you to our most recent SEC filings for a full discussion of our risk factors. So for those of you who don't know Synlogic, we're really at this intersection of two really exciting areas of biology. And we apply synthetic biology-based techniques to probiotic bacteria. And that way we're hoping to create a completely novel treatment modality that allows us to address unmet needs in completely new ways. So we call our therapeutics, or potential therapeutics, synthetic biotics. And that term really underlines this combination of programmable precision genetic engineering with a well-characterized probiotic chassis to create a product that performs a disease-modifying function that's required to either ameliorate or treat a disease. And we believe this platform and these synthetic biotics have potential to be really differentiated drug candidates. We're targeting initially metabolites, where we understand the biology of the underlying diseases. These products would be orally administered. There's no systemic absorption, and therefore it avoids some systemic adverse events. The bacteria are designed not to be colonizing, so once a patient stops taking them, they're cleared from their GI systems. And we believe this platform has potential to address both rare diseases as well as common diseases. Our initial focus is in metabolic disease and immune-based diseases, but we see over time that this is a platform and a technology that could be applied to diseases and therapeutic areas outside of our areas of initial focus. And so this has really led to, over the past 6 years or so, establishment of a really deep and very interesting pipeline with multiple programs that are now in the clinic and clinical development. As I mentioned earlier, our internal focus is on rare and niche metabolic diseases. But we also have some programs in immunology-based diseases, namely inflammatory bowel disease. We have a single target collaboration with Roche to develop an engineered synthetic biotic for the treatment of inflammatory bowel disease, and that collaboration is progressing well. I'm not going to discuss the immunology portfolio today. I'm really going to focus our time speaking about our lead clinical assets. We have 2 products in the clinic for the treatment of PKU. I'll tell you a little bit about those. And then I'll also briefly touch on our 2 additional clinical programs. One is in a disease called homocystinuria, which is another inborn error of metabolism. And then our 3rd clinical program is in a disease called enteric hyperoxaluria. And we're expecting data from all 3 programs this year in 2022. So first of all, to touch on PKU and why we're so excited about the PKU opportunity. Number one, this is a rare disease. These are, most of us who were born after about 1970 have been tested for PKU at birth. So it's part of the heel prick birth testing algorithm for kids. There's about 17,000 patients with PKU here in the US. There's the same again in Europe. And despite the fact that it's a very well understood and well recognized disease, about 75% of patients currently remain untreated with no pharmacotherapy. And the reason for that is the 2 products that are currently approved treatments for PKU continue to leave a substantial number and substantial proportion of patients behind. And we believe that this combination of a de-risked regulatory path based on the precedence of the 2 approved products, as well as this big proportion of patients who currently have no available therapeutic prescription option, is really a very interesting opportunity for us to think about deploying our synthetic biology-based approach. This patient population is just, fantastic to work with. They're very well engaged, well connected, very strong advocacy and community links. And we believe a chronic oral product with no systemic or scary side effects would have a very differentiated clinical profile in this population. And we have established proof of concept, as I'll show you, last year based on our first readout from the Symphony 1 trial. Things are going well, and we're hoping to initiate a phase 3 study in the first half of 2023. So this program is moving along very rapidly. And so just to touch in a little bit more detail on the current unmet need. As I mentioned, 17,000 patients in the US, 75% of whom have no treatment option. And the reason for that is the first treatment that was approved by the FDA is sapropterin or Kuvan. It was a great oral product. Unfortunately, only a small proportion of patients with a more mild type of PKU disease will respond to that product. So for about 20 to 30% of patients, they're evaluated at age 2, whether they respond or not. If you're a responder to Kuvan, generally these patients will stay on Kuvan for life. If you're a non-responder for Kuvan at age 2, there's really no alternative available. You need to maintain this very, very strict diet with exclusion of almost all natural protein from your diet as a way to try and maintain your blood Phe levels in target. The second product that was approved for PKU patients is a product called pegvaliase. It's a enzyme replacement therapy. This is approved for adults with PKU. It has, in patients who can get through the tolerability issues, it can be a very effective product. It is a good treatment. About 1,000 patients with PKU in the US are taking pegvaliase successfully. Unfortunately, not all patients with PKU are suitable for that treatment because of the risks of anaphylaxis and the requirement to take sometimes multiple daily injections, as well as this prolonged dose titration period. So that leaves, a very significant proportion of patients really in need of new treatment options, which we think is an interesting place for us to develop a new approach. We believe our products can be used as a monotherapy in, that dark blue portion of this pie chart. We also believe that there is a proportion of patients who are responders to sapropterin where their blood Phe levels remain above the target range. And we believe that an oral product could be an interesting adjunctive approach for those patients who derive some benefit for Kuvan, but still don't get to where they need to be based on their physician's guidelines. So we think this is an interesting approach. As I mentioned earlier, our product is not absorbed systemically, and I think that's been borne out by the clinical data that we've seen. And it has the convenience of this oral sachet administration that's mixed with 4 ounces of water and consumed by patients. So we think this is a very interesting profile for the patients with PKU who either need a new mono or adjunctive therapy. So as I mentioned earlier, we had some very interesting proof of concept in September of last year. We showed in a study in patients with PKU who had elevated blood Phe levels at baseline that we were able to lower blood Phe in those patients in a clinically meaningful and statistically significant way. The mean change in blood Phe in an all-comers analysis was a 20% lowering, and that's twice the lowering that was seen in the sapropterin pivotal study in the all-comers analysis. So we believe that this is really shaping up to be a potentially compelling product in this indication. What's not shown on this slide is that we collected multiple other mechanistic biomarkers that all very much supported this Phe lowering as being a direct effect of the investigational product. So, all of the arrows were pointing in the right direction, and it was a very robust data set in taking all of the endpoints together. In terms of safety, we've also been very pleased that the product is performing as designed. So it stays in the GI tract, and we've seen no serious adverse events. We've seen no systemic adverse events. Most events are GI in nature and mild to moderate. So patients will often describe change in bowel habit, gassiness, nausea and vomiting. We had one patient discontinue the phase 2 study due to anxiety, which was a pre-existing condition related to that patient's PKU. So in terms of the regulatory path, we believe that this is de-risked on multiple levels from a disease perspective. PKU has 2 approved products. Both products were approved on the basis of blood Phe lowering on a single pivotal trial. So that's a very interesting and important learning for us as we think about planning for a phase 3 program. Our synthetic biotics have a lot of technical attributes that I think lend themselves to the regulatory approval path, and the FDA has a guidance around live bacterial products that we can refer to as we think about our manufacturing process. And then we have 2 drug candidates that are very, very similar. 1618 is our first gen product. 1934 is our second gen product that has 2x the efficacy of 1618. Both are very, very similar. The only difference is a small modification to increase the potency of the enzyme that we've engineered into that bacteria. So that's it for the PKU program. Just to briefly touch on our other programs, homocystinuria is a disease very similar to PKU. It's an inborn error of metabolism. These patients have a defect in the metabolism of methionine, which is another amino acid that results in a buildup of blood homocysteine. Very, very severe disease. These patients can present with strokes at a very young age in their early teens or 20s. No real approved treatment options recently, some protein restriction, very, very challenging, and some historical products that are very limited in terms of side effects, tolerability, and efficacy. That program is currently in IND enabling studies. We have communicated that we're aiming to have a phase 1 proof of mechanism data later this year. And that's obviously a program we'll be moving forward very rapidly if we get the signal we expect to see, given the unmet need in that disease area. Then the third program that will have a readout this year is another metabolic disease, but this time it's not an inherited disease, it's an acquired disease. It's called enteric hyperoxaluria. These are patients that absorb too much oxalate from their diet, which results in elevated urinary oxalate, and these patients can get recurrent stones and eventually kidney failure if this disease is not treated and reversed. We have very nice proof of concept that we shared last year and are now moving forward in a study in patients with Roux-en-Y gastric bypass to evaluate whether we can lower urinary oxalate in that patient group as well. And we'll share data later this year from that study. So a very interesting pipeline of clinical programs with a number of other programs that are moving forward in preclinical research coming along behind. So in terms of the current milestones and runway, we have a number of readouts that are all lining up for the second half of next year. We'll have phase 2 data from our PKU program. We'll have data from our homocystinuria program in healthy volunteers, and we'll have data from the enteric hyperoxaluria program. And then finally, we are making investments and planning to initiate a phase 3 study in the first half of 2023. We're very fortunate, particularly given the market backdrop, to be very well funded. We have a balance sheet of 120 million at the end of the last quarter, with a projected runway into 2024. So sufficient funding to fund all of these milestones that I've just outlined for you. So that's it in terms of my presentation, Keay. I'd welcome any questions that you have or that the audience have on any of the material I've presented.
Well, great, Aoife. Well, to start, let's talk about your platform and your use of synthetic biology. At Chardan, we cover a number of emerging therapeutic modalities, and an important consideration that handicaps early probability of success in the clinic is pursuing validated disease targets. For gene therapies, this means targeting monogenic diseases. For RNAi, it's gain or loss of function mutations. Can you talk about how your initial pursuit of metabolic diseases in which the biology is well validated due to exclusion diets and also having easily accessible biomarkers is analogous?
Yes, that's a great question. It's something every platform company has to go through in terms of the decision making about where to focus initially. And we looked for diseases that really fit in the kind of central part of 4 different circles in a Venn diagram, if you will. Number one, we wanted diseases that were a good fit with our technology. And something that bacteria do very well is scavenge metabolites. So there's a ton of diversity within the bacterial world around your different enzyme pathways. So that was a really natural fit for us in terms of the technology we were developing. You then look at unmet medical need, and I think there's tremendous unmet medical need in some metabolic diseases. And then you look for the biology and the fit with the biology. And what was really important to us early on was that we don't take on too much biology risk. Given that we're developing a new technology, we're learning, where this technology can play, we're establishing the regulatory pathways. So we really looked for diseases where the biology was very well understood. And in our lead diseases in PKU and HCU, they're due to a monogenic defect. The bad actor in terms of the metabolite that accumulates in these patients is well described. There's a biomarker that's followed clinically in these patients. That's a biochemical endpoint. So when patients with PKU go to their physician, they all have a blood Phe measure taken, and there's a target range established for that blood Phe level. And then finally, we know that if you exclude Phe from the diet, you're having a clinically important impact in these patients. So having that kind of validation and having that intersect with our technology, which was very much focused on metabolizing toxins, was just a very, very good fit. And then the fourth component of that kind of Venn diagram with the 4 circles is development feasibility. And you already touched on a couple of really key components there. Being able to follow biomarkers in healthy volunteers in early clinical studies and having good line of sight that those biomarkers will translate to your endpoint is just so, so important for small companies. You really have to prove that you're doing something that's important in a relatively short period of time. And if you can do that in a healthy volunteer couple of cohorts, that will give you tremendous advantages in terms of being able to learn early in your clinical experience, being able to think about finding the right dose, finding the right patient population. And in all of our diseases, there are biochemical, often multiple biochemical endpoints that we can follow. And then the final component of development feasibility is the regulatory path, right? So understanding the regulatory precedent, making sure there's a patient population that can be enrolled in clinical trials. And so when you look at all 4 of those components, you're looking for those diseases that are right at the intersection. And the earlier you are, the tighter you need to be to the central part of that graphic. I think as we start to de-risk our technology, as we start to learn about the clinical experience in PKU, we may take on more biology risk. We may go for, novel things. We may go for, inflammatory bowel disease or more complex diseases where maybe there's a little bit of uncertainty about the metabolic pathways or about the immune pathways and whether they really impact disease outcomes. But I think you kind of have to walk before you run. And for me, the walking component is, kind of sticking pretty closely to low risk biology as we start to think about the initial programs, and then expanding into maybe more higher risk biology as we learn more.
Well, great. And then just a follow-up question on the platform. In your initial indication for PKU, you talked about having 2 candidates. We think of 1618 as version 1.0 and 1934 as version 2.0. Again, using everything you know on your platform, designed to be better. Obviously, we're anxiously awaiting the results from that cohort arm. So 2 questions. 1, how are these strains different? How are they similar? And then number 2, going back to your most recent quarterly call, management did state a precedent of wanting to advance 1934, assuming the data supports it. How would advancing that instead of 1618 in any way affect the path towards starting a phase 3 study?
Yes. So, a little bit of history of how we came to 1934. So initially at the company, 1618 was one of the first strains we developed. We used a version, the kind of the effector piece there, the good stuff is the PAL enzyme that we've engineered into the genome of the bacteria. And so when we designed 1934, we took a PAL enzyme that had been described in the literature, we plugged it into our chassis organism, and that was how we developed that candidate. But we always asked ourselves, did we choose the best one? Is there a better PAL enzyme we could have plugged in? And so in parallel with executing the clinical program, we also had a research initiative going on to look for better versions of PAL. And we deployed a technology called directed evolution, where we basically took our current strain and we looked for mutations in the PAL enzyme that might confer higher activity. And we were very successful in that. Directed evolution, I think, is very powerful technology. We were able to apply some kind of cutting edge techniques to develop what we call a modified PAL. And so it's essentially the exact same enzyme, except for we have 5-point mutations. And those 5-point mutations confer about a 2-fold greater activity on a cell per cell basis. So with the same dose of 1618 and 1934, we consistently get 2 times more activity with 1934. And that's really driven from 5-point mutations. Everything else is the same. So in some ways, it's just like using a higher dose of the same product. We decided to file a separate IND because the products were genetically different. But it's so similar, 99.9% of the genome is the same. It's performing the same conversion. It's doing everything the same as 1618, except for it's doing it 2 times better. So that's kind of the difference between both strains. And as of today, everything we've done for 1618, we've done with 1934, right? We've done the same preclinical models. We've done the same healthy volunteer study. It's now in an arm of the Symphony study. And that's all with the design of being able to move 1934 as quickly as possible into phase 3, should the data that we're currently gathering in Symphony look supportive of that decision. I think something, strange would have to happen for 1934 not to be the candidate, because across every single assay that we've run in the company, be it in vitro in a test tube, in vivo in a preclinical model, or in healthy volunteers, we consistently see the same thing, which is at the same dose, 1934 is consistently 2 times more active compared to 1618. So our assumption is that 1934 is the phase 3 candidate, and that's what we're planning towards. We're just waiting for some more data from the phase 2 study to make sure, and then we'll have a complete matched set of data for both candidates, and I think be able to make the fully informed decision and have that regulatory engagement. That's another step towards starting the phase 3 trial.
Great. So you touched on the commercial market opportunity for PKU. There are 2 approved drugs, Palynziq and Kuvan. So it actually, to your point, it does de-risk a lot of the aspects in terms of certainly the regulatory pathway has been established. There isn't going to be a huge need to educate physicians and patients about the disease and treatment options. But if you're able to get there, let's talk about 2 things. One, the benefit you might show, and let's say it's a 20% reduction, perhaps while good enough for FDA approval, what might that mean in the marketplace?
Yeah. So I think you can think about, there's many ways you can kind of segment the PKU patient opportunity. But we are pursuing initially patients who have a blood Phe level above 600 and bringing them down into the target range. And that's going to have so many improvements for those patients of getting them down closer to the target range. We know that the closer you get to the target range, the better the outcomes are from a cognition, from a clarity, from these patients' ability to kind of keep down a job, have relationships, all those kind of aspects. But what patients really talk about, and where most of the demand is actually coming from, is patients who are on a very tightly controlled diet. And what they really want is to be able to eat more natural protein. Because for patients who are compliant, who have a Phe level in the normal range, they're often doing that at the cost of feeling constantly hungry, unable to eat normal food, relying on formula and mixtures for most of their dietary intake, supplemented by, some lettuce and other low protein foods. Very, very challenging. And those patients just really want to be able to eat more natural protein. So that's kind of another kind of segment. And when you talk to those patients about what would be meaningful for them and what would be important, and we published a survey actually about this, they consistently say that something that would allow them to eat 10 grams more of natural protein would be a miracle. So very small in what would be, nothing for those of us who eat a normal balanced diet would be amazingly meaningful for these patients who are managing a really restricted protein diet. So I think regardless of whether you talk about the uncontrolled patient population or the controlled patient population, I think there's big unmet needs in this disease. And we constantly hear that feedback from patients that, we'd love to get as much as possible additional protein and get the Phe levels as low as we possibly can, which is why we developed 1934. but we constantly hear that something as low as a 20% Phe lowering or something as low as, a 5 to 10 grams additional protein would be really meaningful for these patients. So I think that's how we've been thinking about kind of addressing the unmet needs and what would be kind of a viable product to take forward into commercial.
Okay, and you also touched upon your other two assets, 8802 and enteric hyperoxaluria, 1353 and HCU. What have you learned from your initial development work with 1618 that you've been able to apply to be smarter about how you proceed with 8802 and 1353?
Yeah, so I think there's a ton of learning. So we think about, it starts from the synthetic biology component and really developing the investigational product. And synthetic biology allows us to have reusable parts. So you'll see the same components, the same chassis organism will show up in multiple products, the same promoters will show up in multiple products. So we've established all these tools now, they're on a shelf, we've validated them, so that when we're developing a new strain, we can pull off some of these tools. We already know how they work. FDA has already seen them in a prior product. We have a lot of information about them. So if you look at all of our strains, you'll see there are a lot of kind of common features and themes appear in each one of them. So that's just amazingly efficient from a product, kind of early product development. And then you think about the preclinical path, and what we've been able to establish is by studying the preclinical distribution and toxicology of our lead product, we're now able to apply a lot of those learnings to every subsequent IND. So unless we're introducing something that's bringing in additional risk, we've been able to bypass a lot of the preclinical tox work that would be required, say, if you were developing a small molecule, because these products are so similar from a risk perspective and from a product perspective that we can really shorten the time from lead candidate to IND and dosing in humans. And then when you think about the human experience, we've learned a tremendous amount. We know the dose range where we need to be from a tolerability perspective. We understand the manufacturing component. All of our INDs now are done with the lyophilized product, not necessarily the earlier liquid formulation. We understand how to titrate dose to minimize the GI adverse events. We understand how to escalate dose. So there's been tremendous learnings, I think, across each component that really have generated kind of a flywheel, if you will, so that each product has required fewer investments from the company, shorter time. And I think we've been able to kind of bring higher quality products into the clinic as a result of all of the development work we've done over the past 6 or 7 years.
Okay, great. No surprise, we've been in a tough market condition for biotech and the market in general in the first half of the year. Unfortunately, we've seen a lot of companies in an effort to preserve cash talk about reprioritizing their pipelines, headcount reductions. You guys were very savvy last year. You did a raise in September when you didn't necessarily need to, but it certainly gave you that added buffer. As you touched upon, you have $120 million coming out of the last quarter, get you into 2024. All that being said, what are you doing? What are you thinking on the margin to perhaps be, more conservative if necessary about stretching your cash and not having to make those tough decisions that we've seen other companies have to do?
Yeah, so we're very fortunate. I mean, it could have gone another way. I wish I had the benefit of foresight last year. It really wasn't. It was, I think, good advice and, a good investor support to really fund the company appropriately because of a belief in what we were doing. So we're very happy to have the cash on hand that we have. It covers us well to do all of the assets. But as you point out, our cost of capital has gone up in the last 6 months. So we really made sure that we're looking and examining every dollar that we're spending. Is it a nice to have? Is it a must have? And when we talk about must haves, it's really things that are going to move the needle in terms of getting the clinical data. So, in some cases, it may be prudent to spend more in order to make sure that you're getting that de-risking clinical data, the clinical efficacy data. But we've really taken a fresh look at every single line item in our budget. I think no different to every biotech company. And we've been looking at it and making sure that we're being prudent, that we're really investing in areas that add value. And then revisiting, things that we've already decided to do and maybe looking and seeing, is there a smarter way that we can operate here? is there a way that we can think about getting the same information, maybe in a more creative way? So all those things have been gone on. I think the team and the management team have done a fantastic job. I'm blessed that, our management team, most of them have been through tough periods in biotech, so they know the playbook, which I think has made a huge difference to us and our ability to kind of weather the storm. We have a very experienced team who've been through the ups and downs before. So, it certainly helped from my perspective as CEO when the team had that experience. So, we're hopeful for the data readouts. I think we're in a relatively good position, as you say, relative to peers, and are very excited for what's ahead later this year.
Okay, so final question for you, Aoife. What do you think, and I guess this can kind of serve as a summary, but what do you think that investors really need to appreciate and understand about the Synlogic story to fully get it?
Yeah, I think what we've heard from investors in the past is that we're a science experiment and not a clinical program. I think, we need to re-educate, and say, look, we have 3 clinical programs. These are viable commercial opportunities. These are real products that we're developing. I think some people are kind of enamored with the science, but haven't necessarily appreciated all of the advancements that we've made in the past 2 years around really making these development assets that are going to be a drug someday. And I think refreshing that component of the story has been really important. And I think we're moving ahead. We're determined to initiate a phase 3 study next year if the data continues to be supportive as it has been. And I think the train's leaving the station on the development pipeline. So, certainly as a drug developer, it's a very exciting time to be at the company. And with 3 programs reading out this year, I think it's going to be a very fun time for all of us.
Okay, well, great. Thank you so much for joining us today. And with that, we'll end our session.
Okay.